As we turn our attention to Chapter 17, the last chapter in this semester, we're going to look at the oxidation of amino acids. So up to this point, we focused on how to extract energy from carbohydrates, how to extract energy from fats. Our third source of energy is extracting energy from the amino acids, or proteins themselves. And with this, some of the outcomes that we're going to have is the synthesis of ATP, which is what we want to have happen when we bring in foodstuffs. But also, we're going to produce a waste product that is unique in the form of urea. We will continue to produce CO2 and water as we do oxidation of carbons. In terms of fats, the waste products would be the carbons of the fats get produced as CO2 and water, and then we create ATP. So the urea production, that particular molecule, is associated with the amino acids, and we have to do a whole series of things to look at, and we have to control the concentration of nitrogen in the biological systems. Urea itself is, I believe, the first elucidated pathway in the body. So the one we discovered first. It was a little bit more complex because it is a cyclic pathway, which is really fun to try to figure out if you didn't know that. Quite frankly, we thought about pathways, or synthetic pathways or degradative pathways, as always being linear, and the circular is hard to do because where do you start? You can start at any place on that circle. When we think about protein, there are two different ways in which we degrade proteins. One is the proteins that we eat, and then through normal turnover inside the cells, proteins are synthesized. They have a certain lifetime in which they are functional, and then the body tends to renew them and degrade them. The control of the protein itself inside the cell can also be transitory, in that you get a signal to upregulate a certain set of proteins in your body for a certain molecular condition, and then all of a sudden those particular proteins will go down in concentration because the signal is not there. So sometimes the protein in the cells are at a housekeeping level, always being synthesized, always being degraded, kept at about the same concentration all the time. And then sometimes proteins inside the cell go high and low depending upon signaling. In terms of dietary protein, at dinner time, at lunchtime, at breakfast time, when you eat, you get a large influx. We take care of it then. And then there are times in which you're not eating, and so this is not active. And so you start the digestive process by eating and chewing your food. It comes down into the stomach, and you know the stomach has a very low pH, which can help hydrolyze the protein. Although the pH, even though it's very, very acidic, the protein doesn't stay inside the stomach long enough just for the acidic nature of that compartment to degrade the protein completely. It actually starts, but it really isn't what's really breaking down the proteins themselves. What really happens is that low pH allows the proteins to be denatured or opened up. And so we allow the amino acid chains to be exposed so that we can have enzymes to react with them. Now, there are some enzymes, including pepsin, that is optimized to function at the pH within the stomach compartment. And so it's highly active at a pH of about 2. The acidity comes from a proton pump that is stimulated by the enzyme gastrin, and it pumps in both protons and chloride ions. So essentially you have hydrochloric acid in that compartment, and that's what really acidifies the stomach. And the pepsinogen is the way we control the activity of most proteases. They're synthesized a little bit long, and under certain conditions they're cleaved to be irreversibly activated, converting the pepsinogen into pepsin, the active degradative protein, and then that enzyme goes around and breaks down all proteins it sees, including itself as it gets lower and lower in terms of concentration of protein, and that's how it is turned off or regulated. It will break down itself and then lose its activity. Gastrin also is one of the key molecules that are sent out that give you the hunger response. And so you start breaking, unraveling the protein, breaking them down, but you don't break them completely. You break them into large chunks. They are still long strands of amino acids. You go ahead and then you transition them into the intestine, and the pancreas excretes bicarbonate, which is a base, which then neutralizes the solution. And so we come from very acidic to more neutral conditions once you reach the intestine. The small intestine contains a variety or a whole bunch of proteases that will break at certain amino acids, and we try to get them down into single amino acids as we go through the process. There's also other enzymes in the different compartments as it goes through. And so the work and action of all of these proteases working at different amino acids, breaking usually at the carboxy end. Once it's processed by all of these proteases, we're getting single amino acids. And most of the time, all of our foods are absorbed in what would be the monomeric form of whatever polymer we have, whether it be sugar, whether it be protein, or we break them into the components of fat. They are brought through the membrane of the intestine as the single units for further processing. In turnover, there are two ways proteins are destroyed into amino acids or hydrolyzed into amino acids. One is through the lysozyme, which has an acidic environment, and proteins that will add water, breaking the amino acids down into single amino acids. The other way that proteins are identified is over time. Proteins will have a small protein added to them, and it's time dependent. The protein that's added to other proteins is the protein ubiquinin. And so over a certain period of time, it's roaming around doing its thing, and ubiquinin gets added. Okay? And then it goes around for a long time, and then another ubiquinin is added. It's almost kind of like its age. And so the more ubiquinins you have, the older it is. Once you get to a certain number of ubiquinins, one doesn't tag it, but multiples tag it to bind to the proteasome. And the proteasome is ATP dependent. It feeds the protein into the complex, and there are protease active sites inside there, and small chains of or blocks of amino acids are released, which then can be further digested. The ubiquination, actually has three proteins that work together. The ubiquinin is 76 amino acids long. Ubiquinin gets its name because it is ubiquitous within the biological system. That protein is everywhere. And it will connect to the proteins over time, and what it does, it actually adds to the proteins one at a time until you get to at least 4, which tags it for targeting to the proteasome. So if you look at enzyme 1, the ubiquinin actually is activated by being covalently linked to a thiol, to a cysteine. It gets carried from enzyme 1 to enzyme 2, and it gets transferred to enzyme 2. Enzyme 2 binds to enzyme 3, which enzyme 3 and 2 create a unique binding site for all sorts of different proteins. And so the target protein there in number 4, the stage here, whatever is going to be ubiquinated, all right, binds to this complex, and the ubiquinin is often attached to the N-terminus. And it could attach one and be roaming around for quite a long time before it encounters this complex again, and over time then it will have enough to bring it to the proteasome. The amino acids, unlike sugars or unlike fats, okay, we have no storage molecule or polymer of amino acids. I can put sugars together and make glycogen, okay, and then I can degrade them and have glucose back. I can take fats and actually compartmentalize them, send them to adipose tissue, put them right into fat cells if I wanted. The amino acids, on the other hand, if they are brought in, the amino acids have to be connected to a transfer RNA ready to be synthesized into a new protein that has to be signaled to be upregulated and synthesized. But if you aren't synthesizing proteins, then the amino acids must be degraded. And so I don't have this store, or I can't just put a polymer of amino acids together to hold it off to be used later. Now, fortunately, within the biological system, we have a lot of muscle, a lot of protein that is available, and so we have a source of amino acids that can be used. But if you eat a large meal of amino acids, then most of it will be degraded. Very little of it will be used for synthesis at that moment in time. This is part of the idea behind the Atkins diet, in which if you actually have lots and lots of amino acids, most of them are degraded and placed into the citric acid cycle. To continue to allow the citric acid cycle to go, and if you have this large influx of amino acids that must be broken down, you need acetyl-CoA. What the body will do in the Atkins diet, if you aren't bringing in carbohydrates to make the acetyl-CoA, you will upregulate using fat. And so you must break down all that amino acids, you will extract the fat, and you will lose the weight. And so with the Atkins diet, you can eat as much protein as you want, just about, and you can even have fatty proteins that are coming in because those fats will be broken down along with the other fat to be mobilized. Now here is the problem when we do break down amino acids. The caution is when you release the amine, which has to be taken off the amino acid, the ammonium that is released, very small concentrations are toxic. So when we're degrading proteins, we have to really control where that amine goes, and it can't be just left free to roam around the solution. We have to take it, place it into a molecule that is non-toxic to us so that we can excrete it, and that molecule will be most likely the urea. Now, the amino acids can be transported to the liver, okay, and they convert mainly a lot of times in the liver, and what they do is they have a two-part degradative process. One part is to take out the amine, and I can do two different things. One, I can create urea, and that's a total waste product. But the fact that nitrogen is found in nature most often as the gas in triple bonded to N, it is a molecule that is in great demand for growth. It is very hard to break that triple bond. We can't do it. Plants can't do it, but the microorganisms on in the roots of plants can fix or break those nitrogens into single nitrogens that are reactive species. The other place that breaks down nitrogen is lightning. That's enough energy to actually break down the amine. If by chance we get rid of the amine as urea, it is very costly within the biome to get more nitrogen back, and so the body attempts to take the nitrogen and recycle it to create other amino acids or nucleotides or other biological entities if it can. And so there's a balance between trying to find a place to place the amines in synthetic pathways. If all of those are full, then we go through the urea cycle. Secondary to that, once I take care of the amine, or that process is going on the other end, we're going to take the carbon skeleton and we're going to degrade those. And what we're going to do as we look at each amino acid, we're going to see what carbon chain goes into which of the different pathways that we already know. So we'll do some mechanisms until we get to a molecule that's in one pathway, and then from that I won't teach you anymore because I assume you know glycolysis and citric acid cycle and things of that nature. All right? And so we're going to take care of both of those parts. In processing the amine itself, okay, the amines tend to go to one or two entities. In the degradation of the amine to urea, most amines gets transferred to the amino acid glutamate. And so glutamate will pick up the amines, and I'll have a very large glutamate concentration. I can also shift into glutamine. I'll show you that in a moment. If you're breaking down muscle the protein there into energy sources, the amines are placed into alanine. And the alanine then gets excreted out of the muscle into the bloodstream and accepted or taken up by the liver. And so the amines are transferred through alanine to get to the liver from muscle cells, and we see that quite often. And so there is a cycle in which you are breaking down amino acids in the muscle cell itself. You get protein degradation, amino acids. Okay, I will go through glutamate to put the amine into alanine, and that alanine will then go to the liver. And from that point on, it can go through the urea cycle to be excreted as urea, and then I can synthesize the carrying molecule, which is pyruvate, back into glucose. And so if by chance you eat protein, okay, we take every amino acid and we transfer the amine into glutamate by reacting it with alpha-ketoglutarate, and I will have then the carbon chain that I can degrade. Cellular proteins are also broken down. The amine is placed into alpha-ketoglutarate. If you are degrading at a slow rate, the glutamate concentration is enough to hold on to the amine. If you're producing a lot of amines, what you can do is glutamate is a carrier that can carry one, or if you react it with the R group, you get glutamine, which is a much larger concentration of amine into a carbon source. And so you can shift between these two intermediates, or those two amino acids, as buffers of amine before it's released. In terms of actually getting rid of it, the glutamate will react to release the free amine, and again, this can't go over a certain concentration, which then gets produced into urea. The alanine is what's coming from the muscle cell if I'm breaking down protein in muscle, and we can also release glutamine, which is not shown, from the muscle itself, and allow the amines to be carried to the liver there. So, in the reaction to handle the amine, and we'll take a look at the amine first, we use an enzyme called aminotransferase. And so the general reaction is whatever amino acid you want to take and alpha-ketoglutarate. What you will produce is what's called an alpha-keto acid and glutamate. Now, what in the world is an alpha-keto acid? An acid group with a ketone. Alpha-ketoglutarate is an alpha-keto acid. If you change and take this amine off and give it here, the nitrogen to carbon bond becomes a ketone as part of the mechanism. And what's left is an acid and a keto, an alpha keto acid. So all amino acids get converted into different alpha keto acids. All right, so that's the generalized. And so if I want to transfer aspartate to alpha ketoglutarate, we use aspartate aminotransferase. And if it's alanine, we use alanine aminotransferase. And there are 18 of these. Okay? In animals, there are 2 of the amino acids that are not seen. There's not an aminotransferase for lysine and threonine. There's other ways that we extract those amines. But most of them will have their own aminotransferase and give initially the amine to glutamate. Then glutamate takes it on to process. What this does is it simplifies the degradative process. I don't have to have 20 separate degradative processes for each amino acid. I take all these amines, put it on glutamate, really, and then I take glutamate and take the amine off. So it simplifies the process. Aminotransferase has a cofactor that is used called PLP. When have we seen PLP before as a cofactor? We have seen one enzyme in this semester using PLP. No, it wasn't in the pyruvate dehydrogenase complex. But that's a good guess because there was a lot of things going on there. That was TPP. Okay, it is another B vitamin derivative, but it was another process.
Is it the one that that penicillin is like a suicide inhibitor too?
No. That's also a really good guess, but no, it wasn't there. All right, I'll tell you where. It's in the enzyme glycogen phosphorylase. In that reaction, what was important was in the phosphorylase is this phosphate coordinated the phosphate that was coming in to break down the glucose. So this was the important functional group in glycogen degradation. And the aminotransferase, this has nothing to do with the reaction. It's going to use a totally different part of the cofactor. And what we're going to do is we're going to use this ring and this ketone as part of the reaction mechanism. So this is kind of interesting in that we use the same cofactor, but we use it in different ways. We saw TPP before, and we've seen it used in a couple of reactions, but the TPP was always used as a nucleophile, and it was used exactly the same in different enzymes. Here we're using a cofactor in 2 separate different ways. You can see what the vitamin B6 that we eat is the alcohol, and then the oxidized aldehyde, and the phosphorylated version is the cofactor. The PLP is actually linked to a lysine that's in the active site. And so here's the ring of the PLP. That's that phosphate. Okay. And what we have is a Schiff's base that holds between the lysine and the PLP. Now, if I say Schiff's base, what is the structure that you think about? Because Schiff's base happens in biological systems all the time. What is the functional group if I talk about a Schiff's base? What is it? No, it's not a metal center. It is this, a carbon double bonded to a nitrogen, and normally it has a positive charge. It is formed from the reaction of a carbonyl and an amine. Most of the time, the amine is a lysine chain in the protein. And the carbonyl, whether it be an aldehyde or a ketone, comes from an organic cofactor. It's a reversible reaction with the addition of water to break it or taking away water to make it. So it's reversible. And it's a way to covalently link something together that creates a very, very strong bond. Okay, so a cofactor is bound into the enzyme and actually physically connected. But we've also seen CHYS bases used as a mechanism like in the aldolase or the transaldolase, such that a leaving group can be created because I have a kick up of electrons to neutralize that positive charge. So CHYS bases are used quite often, and it's a reaction of a ketone and an amine, where it once was a carbon double bonded to an oxygen becomes carbon double bonded to a nitrogen. Okay, and so if you see that, you need to understand that particular or what that functional group is. And so here is a generalized amino acid coming in, and we have the enzyme that has that CHYS base, the catalytic subunit, the cofactor, inside the active site. What will happen initially is that the amino acid and the CHYS base will react. Now, I'm gonna show this mechanism. I'll let you jump in when we start degrading amino acids. I will not forget. Even tomorrow, if we don't get to something, you're on deck. Okay. But I'm going to show you what happens here. Okay. The amino acids comes into the enzyme, and it is normally protonated in the form of NH3 with a positive state. That's because of the pKa of the solution and the pKa of the nitrogen and the pH of the solution. If you can see, this nitrogen is in the amino acid. That's the nitrogen. It has to connect to the carbon that was connected to the nitrogen of the lysine. And so I have to attack it at this carbon so that that carbon has one nitrogen to the lysine and now one nitrogen to the incoming. And so that is not a B. There's a B. The base will take this. I will attack it here. And then, based upon the protonation state here, what I'm going to do is I'm just going to take the pair of electrons and put them on nitrogen to neutralize the positive charge on the lysine's nitrogen. All right. And the next step, what I'm going to do is now have a CHYS base between the carbon of the cofactor and the nitrogen of the amino acid. The lysine is the leaving group. And so to get there, this nitrogen has one hydroxide, this nitrogen has two. Let's take one away. In that, the product has a double bond, so I will take the pair of electrons that are here and I'll form that double bond. The nitrogen's positive charge still is there. And then now I have that carbon with a double bond. I see that. And then I'm missing a bond. That bond missing is this lysine. And so this pair of electrons will place its electrons onto that nitrogen to form the, oh, not an extra electron, to form that lone pair that sits there. So what I've done is I've substituted the cofactor that was once bound to the enzyme, now bound to my amino acid. Why I place that cofactor onto the amino acid, in some way, what I need to do is to get this bond broken. I need to remove that nitrogen. So some way, because this ring system is here, it's going to allow me to break that bond. That's my goal. Takes a couple steps to get to it. All right. What's going to happen is I'm going to allow some reactions to occur in the ring, and this is kind of interesting because the reaction is very far removed in a lot of movements. If I look right here, this carbon that has the acid in the R group and the nitrogen is missing what in the original? That proton, right? And so this proton has to be deprotonated. Now, the cofactor makes it acidic because if I take that off, these electrons can go there, these electrons can go there, these electrons can go there, and these electrons can be on the nitrogen, making it neutral. So that whole series of resonance forms makes that normally the hydrogen on the alpha carbon is not acidic. By putting on the cofactor, it is now acidic and can be deprotonated. Now, that's great in terms of when I started, I had a charged cofactor that was actually 2+ with, it's 1+. You've got nitrogen and nitrogen, that's 2+, and acid, that's a 1 negative. After the deprotonation, I'm neutral. Okay? That seems wonderful in terms of stabilization. Oh wait, I take that back. You also have that negative oxygen, but that's in a hydrogen bond, so that stabilizes it. But there's a problem. That ring movement, what did I destroy by putting the electron pair there? Resonance. What special type of resonance in that ring system? An aromatic ring, which is very stable. So this, although it's a resonance form, it doesn't like to stay around very long, and it will reform its aromatic ring. But if I went all the way back up to where I once was, it's not productive. In this case, what happens is I pick up an additional hydrogen at this carbon, which is this one. And so after the reaction, there is an available proton that this can react with, which wasn't there before. It wasn't optimized before. All right, I create back the aromatic ring, very stable. I now have a carbon that has more hydrogens at this position, and I have a Schiff's base right between, once was the alpha carbon of an amino acid and the nitrogen of the cofactor. All right, what I didn't show and I needed to show is the next reaction has water. And so a base will take the water, the electrons will attack to create the hydroxide, and the electrons will pick up a proton such that this nitrogen at the beginning has 1 hydrogen, it now has 2, and the carbon has a hydroxide attached to it at the alpha carbon. This last step in this half of the reaction takes off this, puts a double bond, and what leaves is the nitrogen. Effectively, all of the carbons of the original amino acid fall off with once was here a hydrogen now has a double bond oxygen, and once was there attached was a nitrogen. Here's the nitrogen of the original amino acid sitting on this cofactor. I've taken the nitrogen away from our amino acid. This is half of the reaction. Because an aminotransferase, if you go back, it took off the amine from one thing and it gave it to another. Okay? And so I have just shown the taking the amine off. And this can be whatever, the alpha keto acid is specific to whatever amino acid came in. We've shown up to this point, half of it, where the amine is sitting here. Now, an alpha keto acid can come in. Most often, it is alpha ketoglutarate, although others can fit in there. And then I really do exactly mechanistically the reverse steps where the nitrogen attacks at this carbon. I go through all of the steps backwards, and the leaving group will be a different amino acid, normally glutamate, and what happens is the cofactor gets attached back to the lysine to be the same structure as it was in the beginning. So it's a two-step process. I bring in the first amino acid, I do a bunch of reactions, take the amine. Let that carbon go out, bring in another, usually alpha ketoglutarate, react it with the nitrogen, and after everything's done, a new amino acid roams out, most of the time glutamate. And I'm ready for another round. Complex reaction, but this has to run a lot when we are degrading amino acids.